Hey guys, welcome to the last topic in chapter 15. This is going to talk about immunologic methods. We introduced this in topic 1, but we're going to go into this in more detail today. And this one's going to be the longest of our 4 topics, so just be prepared for that. It's not super long, but just be aware that it's got a lot of things in it. So immunologic assays are just as important as the phenotypic ones because these are the backbone of what you see in a hospital lab. So it's really important that you understand how these tools are used and when they are used. Here's our topic objectives. As always, if you have any questions, please let me know. These ones you may or may not be very familiar with, so you may have a lot more questions about them. So it's totally okay. Just let me know, office hours, in class, anytime. Just let me know if you have any questions. So there's a general principle of all immunologic assays. The antibodies are both specific and sensitive, meaning that they can determine small volumes of things and they only will match to certain things. Using that information, we can answer 2 main questions. What does the patient's antibodies what are the patient's antibodies specific for? So what are they, either do they have immunity for before, or what potentially are they sick with now? As well as we can use antibodies that have been previously extracted to determine what identity an unknown pathogen has. So there's 2 different keys to this. It's really important to understand that antibodies are really the key to most of these immunological assays. And I'm not going to go through all the immunological assays that are in your book. I'm just picking out the big ones for you. But if you have questions about any of the ones that they briefly mentioned, please let me know. So the first test we're going to talk about is an agglutination precipitation test. What this does is it takes antibodies, and what we'll do is we'll dilute out samples from a patient in a controlled manner, so we know exactly what we're talking about for dilution, and then we place it in the presence of cells. The antibodies, if they're specific for those type of cells, will then pellet in the bottom of the well. This creates this agglutination response, and what that does then is it tells us that they're positive for that sample. If it doesn't, then we see this kind of diffuse pattern in the bottom of the well. And so this allows us to not only know what antibodies a person has, but also what their titer is, so what the level of antibodies they have is. So it's a really nice test in that way. And it's really simple, can be done really quickly, and it can be used in a variety of applications. Another assay that's used in the lab is Western blotting. What we do with this is we take proteins from a sample, usually these are antigen-specific proteins, and we separate them on strips using electrophoresis, so the electricity will pull them apart from each other. We then can stain them with either specific antibodies or colorimetric and match up banding patterns. And this allows us to find out if somebody is positive for whatever we're testing for. In the example from the picture, this is HIV. This is really a useful tool, but it's not something that can be done overnight. Like the agglutination is really easy to do and can be transferred to most tests or to most pathogens, whereas Western blots have to be figured out. We need to know what banding pattern we're looking for. We need to develop positive controls. We need to get the antibodies working out right. So while they're really useful, they're not something that can just be easily maneuvered. They have to be researched and developed before they can be used in a hospital setting. Another technique is immunofluorescence. What we do with this is we take a sample from the patient, we then place it in the presence of antibodies that are specific for the pathogen we're suspecting, and that we then, and these antibodies have fluorescent tags on them, and so we can then measure the amount of fluorescence that we see in the sample to determine if the sample is positive or not. Note that in all of these ones we're talking about, we need to know what we're looking for in advance. These are, the antibodies would then also give us a negative, but if you didn't suspect somebody of syphilis, you wouldn't necessarily do this test for syphilis. You would then do it for whatever you're looking for. So it's important to have some sense of what the symptoms and the signs of the patient are telling us as far as what testing goes. And that's usually what the doctor or the nurse works out before the hospital lab. The hospital lab usually just completes the testing or serves as a expert to provide some sense of what tests should be ordered. So if you've taken A and P, you've done this experiment in the class, but if not, then that's fine. We're going to go through it still. But this is called ELISA, which is an enzyme-linked immunosorbent assay. If you've ever seen a pregnancy test or anything that's a strip test, this is an ELISA as well. In the lab, what we do is we take these wells of these plates and we coat them with either an antigen or an antibody that is specific for something. We then will place the patient's samples within that, and the antibodies will bind to them, or the antigen, depending if we coat it with an antibody, will put an antigen present. If we code it with an antigen, we'll put an antibody present because that's how they work. And then we will put in a colorimetric tag afterwards. So if it's bound, it'll turn a color. This allows us to then have a positive indicator, and you can see that in the bottom of the plate. The wells that are yellow are positive, the wells that are clear are not. And so this allows us, once again, it's kind of like the agglutination, to see that we can either do this in a series of dilutions to get a titer, or we can just do it at a set number to give us a yes or no answer. All these serology tests are really important for viral infections. As you notice in lab, we don't ever culture viruses in your guys' microlab, and that's because viruses are really difficult to culture. So serology testing is really what we're looking for most of the time, because this gives us a better answer than trying to detect very small amounts of virus. And so here is an image where you can see how these different assays are applied for virology testing. But as you know from your own common experiences, that we don't generally test for viruses unless we're looking for something that is either communicable, such as herpes or HIV, or something that's even deadly. So that's when we would pull out those tests. If you have the cold, you don't generally get these tests unless we're worried about a specific type of flu. So let me know if you have any questions about these. Please review your topics, your topic objectives, and let me know if you have any questions about that. I know these are maybe new to you, so that's fine too. Just let me know if you want to go through them again. I went over them pretty briefly here, but there'll be videos in Blackboard for you as well.